so that we can have that class. So since this is the last class, I thought I'll add sort of two options. One is to talk about one of the research topics in the lab, that is neurovascular coupling. But then again, I said I'll be too detailed and too specific, and everyone may not be interested in that specific topic. So I thought of giving an overview of all the activities in the lab, and if anybody is interested, they can come for internships and things like that. So, like I already mentioned once, one of the objectives or philosophies of lab is to come up with a simplified brain theory, so that you can explain more phenomena, and you can also, more importantly, understand diseases better, have a greater insight, and develop treatments based on such fundamental theory. So, as we see now, there's a lot of data that is being accumulated because of the availability of sophisticated techniques in neuroscience at molecular, cellular, systems, and behavioral levels. But there is no commensurate development of theory, as many people have commented. For example, Ramchandran says we are at a Faraday stage in neuroscience. Patricia Churchland, a neurophilosopher and neuroscientist, she says there are no good neural theories. There is a patchwork of theories. And part of the reason is because of the reductionism, that people try to explain all phenomena in terms of the smallest constituents, the neurons or even molecules. That's unnecessary, and I think I gave the example of fluid flow. That is, to describe fluid flow, you invoke an abstraction called fluid and write the dynamics in terms of that fluid and not in terms of molecules. So that's why you're able to understand this system. So because of this problem, there's disproportionate development. There's a lot of data, but at systems level, at large scale, which is where things begin to matter, this consequence of this kind of approach, reductionist approach, will be seen, right, at systems level. At systems level, you have very poor understanding of what's happening. And therefore, the diseases which operate at systems level are also poorly understood, and therefore they are poorly treated. So these are all connected. All these factors are connected. So therefore, there is a need to develop simplified models embodying the right abstraction and the way just as it is done in physics and engineering. If you do that, we strongly believe that neuroscience will see a revolution, and you will see lots of applications, not only in clinical neuroscience, that is neurology and neuropsychiatry. These ideas will also percolate and and inform and illumine machine learning, deep learning, and all these AI-related areas. So to that end, we want to develop a large-scale brain model like this. It's a reduced brain model having maybe about 10 million neurons, not the whole 100 billion, which will have the sensory inputs, motor outputs, the two joining at this decision-making centers of prefrontal cortex, and then the three major lobes, cerebellum, basal ganglia, and hippocampus. So in this, there's already a lot of progress made in terms of visual processing. Sound a little lesser, touch is also a little lesser when I'm talking about modeling. But when these sensory streams, as they climb up, they start merging. Like I was saying yesterday, you generate all these higher level representations of sensory input, or multimodal representation, that is sound and touch vision and touch combined, sound and touch combined, these kinds of representations. And these representations slowly lead to become foundation for our representations of language and space. These are two things that brain quickly develops after some basic processing of sensory information. And it's these higher level representations that are then used for decision making in prefrontal. Right, and on the other side, we have seen there are three motor streams. So the musculoskeletal, that is everything that you do with your skeletal muscle. Then the speech, your larynx and jaws, and that whole articulatory apparatus, that is one thing. And then your frontal eye fields and supplementary eye fields, all these cortical areas which control your eye movements. These are three major motor streams controlling voluntary movement. So our approach to this construction of large-scale brain model is not just to take a huge model with 10 million neurons, 100 billion neurons, and run it on a supercomputer and show that it works. Because that kind of a thing has been done. For example, Izakevich and group have simulated a 100 billion neuron network model of Izakevich neurons, and shown that okay, we can do it. We have enough computational power to do it. We have in this case, which neuron model is small enough, is simple enough to do it. But the point is, what is the point? I mean, what have you demonstrated really? Do you now understand how cerebellum works, or what are the representations of the insular cortex, right, etc., etc. So that kind of just a gross network model, which is uh, where the only virtue is its size, is of no use. You need to have a functional model. The model should have representations of all the major brain structures and should be explain their function. Right? It should, once you have a model like that, you should understand how does hippocampus work, how does basal ganglia work, and how do they all work together, and also explain brain's activities in at least, lot of domains. It's like as good as at least a prototype of the real brain. So that kind of model is still a far cry. We are quite a long way away from that. The best example of that kind of model is a model by Chris Elias Smith. This is already a few years old, maybe at least 5-6 years old. Chris Elias Smith at University of Waterloo. So they have something called a neuroengineering framework, and with that, at that time they developed a model of they call it a whole brain model. It does some visual and motor processing, but it has a visual hierarchy and a motor hierarchy, some components of reward processing, some components of working memory. So it's a reasonable, pretty good model. It has about, I think, 2.3 million neurons, which that was considered one of the, or maybe the largest functional brain model at that time. And that was published in Science. So it really made, got some attention. So, but we need to go beyond. We need to go further and actually incorporate, right? Have representations, accommodate all the major brain structures, and it should be like really a cartoon model of the brain, right? And we are not yet there. So we decided we'll model different functions, different parts of the brain, and then as these models mature, we'll put them together and assemble a whole brain model. And that's the approach we wanted to take. And here, of course, there is also some. It's not that we understand all the components; you just have to implement them, write the code in Python or something, and put it together. It's not like that. We're hardly there. We don't even understand the functions of the individual components. So you can't just write code and assemble all this stuff. So a lot of research has to be done even at the level of fundamental components. And also there is a lot of research and rethinking that has to be done about what is the right representation. And should we work in terms of spiking neurons, should we use red-coded neurons or something else? So all these, a lot of fundamental questions that have to be revisited if you want to create this kind of a brain model. So with all these considerations in mind, we kind of opened up the battle on many fronts and began to work on many aspects of brain function. You see, so the lab is divided into teams. Each team works on one focused topic and try to do everything around that topic. So we have a team on stroke and rehabilitation, another team on deep vision, but deep vision team is kind of dissolved or dissolving because of lack of people. And neurovascular coupling, then neural oscillators, spatial navigation, basal ganglia, and Parkinson's disease. There's a couple of more teams which I'm not showing here. For example, there's another new team on reading and writing, which is also expanding. So there are quite a few teams. Some of the old students. So this is a couple of books that came out of the lab. Collaborators that will teach all this. Yeah, so let us talk about some of these things. Let's give a quick overview. So basal ganglia, so I gave a research talk on basal ganglia work on in last week sometime. Some of you might have attended. But quickly to recap, basal ganglia is a deep brain circuit, a subcortical circuit, having about 7 nuclei or modules. Right, these modules are shown in a simple block diagram like this. So this system receives input from the cortex and returns output back to the cortex. So it forms a loop between. So it forms a cortico-basal ganglia loop, because input from cortex comes here and goes through this network and goes back to the cortex. So this stuff is the basal ganglia circuit. So input from the cortex goes to the input port of basal ganglia, this striatum. Then output from the basal ganglia goes out through two output ports, GPI and SNR, and from there it goes to thalamus and then to cortex. So within the basal ganglia, information goes through two major pathways. So in fact, there are a few more, but anyway, this is a traditional picture. So there is a short pathway from striatum to GPI/SNR. So this is called a direct pathway because obviously it's a direct connection from here to here. And there's a longer route from striatum to GPE to STN to GPI. So since it's a roundabout route, it's called the indirect pathway. There is one more pathway where the cortex and direct inputs to STN that was discovered subsequently. So that's called the hyperdirect pathway for lack of terminology. There's also another connection from GPE to GPI, so that's not given any name to my knowledge. Basically, in the brain, there are anyway a lot more connections than we would like, but anyway, so that's a fact. So the most important thing is there is this set of nuclei, so a set of neurons in this nucleus called substance nigra pars compacta. This way there are kind of cells which release a substance called dopamine. So in Parkinson's condition, these cells die and we don't know why they die. We have some theories, but we don't have a final answer. An answer that is good enough that you can use that knowledge to arrest the death of these cells. But these cells die, and therefore it is because of that one small cause. Right, you see devastating symptoms in this disease. So PD or Parkinson's disease is a progressive neurodegenerative disorder. It's neurodegenerative because the cells degenerate in essence and keep dying. It's progressive because once that disease sets in, the cells start dying. There's no looking back. They keep on dying. So pathology is loss of, like I said, dopamine cells in substance nigra. The four major symptoms, what are called the cardinal symptoms. These are tremor, rigidity, postural abnormalities. So they say that they walk with a stoop. And then bradykinesia refers to brady means slow and kinesia is movement. They move slowly. So in fact, the slowness of movement also seems to be reflected in the slowness of thinking in these patients. So there is also a term called bradyphrenia, so slowness of thinking. Since in fact in extreme cases, it's not even slowness of thinking, it's almost stop thinking, just as they have difficulty even moving. They seem to have difficulty even thinking and willing, that is expressing your willpower. So one of the symptoms of PD is apathy. Apathy is lack of having no interest in anything. These guys can just sit there in the front porch all day long, not doing anything, not wanting to do anything. Right, that's one serious condition. So they have because of so thing is basal ganglia is important for expressing your willed action when you make a voluntary movement. Right, I don't think I mentioned before, but so basically in the motor cortex or in the motor cortical areas, there are several areas, and there are two kinds of movements where even voluntary movements are described. In one kind of voluntary movement, it is more stimulus driven, like when you are catching a ball, right? That the ball is coming towards you, and to meet the ball, right, you move your hand in certain fashion. So your hand movement, although you voluntarily give it a permission, your actual movement is guided by the ball itself in a way, indirectly. But when you are just sitting quietly and lifting your hand, or you want to get up, that intention has come from inside. It's not being driven by outside stimuli. So in the motor cortex, these two are distinguished because in case of internally driven movement, it is the supplementary motor area which becomes very well activated. In case of externally driven movement, it's the premotor area which is activated. So, so in the will disorder, the SMA function is more disturbed, and SMA and basal ganglia are closely coupled. So anyway, so they have. So thing is, because of loss of cells here, so there are all the four major domains of function of brain are affected. The four major domains are sensory motor function, cognitive function, that is memory, sequence processing, decision making, all those things. Then affective function, that is emotional function. Then autonomic function, that is a control of the internal organs. All these four domains are affected. I mean, that's pretty much everything that the brain does is affected just because of loss of cells in this tiny part of the brain, which might have maybe and a few lakh to 1 million neurons or something like that. It's a very small structure. So in motor domain alone, there are so many modalities are affected: hand movements, eye movements, speech, posture, even leg movement. Its gait is also affected. So on the right side here, you see handwriting between an elderly normal patient and a normal subject and a PD patient. See that PD patient writes small letters. So the classical picture of basal ganglia function, if you look at a textbook or look at some of the older papers, will look something like this. So the direct pathway, when it is activated, like we know that it facilitates movement. So that is it makes you go. Therefore, it is called the go pathway. And the indirect pathway, when it is activated, it inhibits movement. So therefore, it is called the no-go pathway. And this switching between go and no-go can be affected by dopamine, which is released by this as SNC structure onto striatum. So what happens is that the striatum is not shown explicitly in this picture, but so when dopamine is released into striatum, when striatal dopamine is in high levels, the cellular circuitry within striatum is such that it will think of dopamine as controlling a valve that controls information traffic in striatum. So when dopamine levels are high, the valve opens in such a way that the information input from the cortex goes straight through the direct pathway. When dopamine levels are low, valve opens in a different way and routes the information coming from the cortex via the indirect pathway. So therefore, which means right when dopamine is high, it facilitates go pathway, so movement is enabled or facilitated. When dopamine is low, movement is blocked because no go pathway inhibits movement. So therefore, you can account for one of the basic features of Parkinson, because when dopamine is high as a normal subject, person can move. When dopamine is low as in a PD patient, there is difficulty in movement. So we visited this problem, and after doing some modeling work, we kind of propose that in the indirect pathway, the main function is not only no-go, it also does something called exploration, right? And therefore, you need to call this not you should not describe this basically into this no-go dichotomy, but you need to describe it in terms of go, explore, and no-go. That is, at high levels of dopamine, it is go, low levels, it's no-go. But at intermediate levels, it shows the ability to explore the space of action. So let me see, right? So we have, that's the main proposal, and then I described all this in more detail in that other talk, which is recorded. If you're interested, I can send you the recording. So we published about 35 journal papers over the last 15 years and worked on a wide variety of uh, topics in basal ganglia, right? Spatial navigation, eye movement generation, deep brain stimulation, bird sound, because birds also have a kind of a homologue of basal ganglia. So we showed how that works. Then risk-based decision making. How is gait affected? Why do PD patients freeze when they walk? Right, and how do we make reaching movements? How are the reaching movements affected in PD? And how is handwriting affected in PD? So quite a few things, and which were all summarized in this book we published a few years ago. So more recently, another student of mine, Vignan, did his PhD on this problem. That is, so until then, in our modeling efforts, we assume that the cells in SNC die, and then we try to work out all the symptoms. Now, if these cells die, why do we have all these symptoms? That was what we addressed until then. But in Vignan's thesis, he said, okay, why are these cells dying in the first place? So there was a couple of people who have hypothesized that the underlying cause of the cell death in PD is loss of metabolic deficiency, insufficient energy delivery to these cells, because these cells, the SNC cells, are form a important network hub in basal ganglia. It's a small structure, but it receives input from many sources and also sends output to many sources. So like because it's a neuromotor system, like what we have seen yesterday. The neuromotor systems have very rich projections to many parts of the brain, and so they have large and complex harbors. So the energy demands are very high, right? So because So any inefficiency in energy delivery, so energy comes from the blood supply in the form of glucose and oxygen. Any insufficiency there will start hitting these cells first because they have very high demands, and neurons cannot tolerate long durations of metabolic insufficiency. So Vignan developed a series of models at multiple levels, at intracellular, modeling intracellular process, network level, and single cell level, and so on. At all the levels, what he has shown is that if you squeeze the energy supply, the system will start showing up some of the PD symptoms. Right inside the cell, it shows phenomena like calcium elevation, ROS production, reaction oxygen, reactive oxygen species production, and alpha-synuclein aggregation. So all these things are observed within the cell, and at the network level, you can see why the SNC cells are dying, and also this death form because these cells form some kind of a positive feedback loop with other basal structures like STN, subthalamic nucleus, and striatum. So therefore, what happens is if there is an initial loss of cells in SNC for whatever reason, right, the way things, the signaling changes within these networks is such that that initial loss is amplified further, and there's a greater tendency to die, right, once there is a initial loss. So there's a kind of a positive feedback loop happening, and that is a so that corresponds, that seems to describe well why there is neurodegeneration. That is, once you have initial loss, the cells keep dying in the real patient. So our models seem to they are indicating that why they keep dying once there is initial loss. So our long-term goal is to the way PD diagnostics and treatment occurs right now. This diagnosis is based on a scale called UPDRS scale or score. So here the clinician observes the patient, the way the patient moves and walks and all that, and makes some qualitative observations and gives it some scores. And so the clinician then plans at the treatment protocol based on those qualitative scores, and they're quantitative but based on pure observation, not based on any quantitative measurement. So we want to change this by having a bunch of clinical measurements. Like for example, you can record the patient's speech, you can record eye movements, you can make the patient perform some gambling, like a decision-making task. This is a standard task called Iowa gambling task, which is used as a kind of a benchmark for decision-making in psychology studies, world over. Then you can make reaching movements. You can give handwriting samples. You can measure gait. What is interesting is all these things can be measured simply using a smartphone. In a very low cost way, you can get all this data. Then feed this data into our model, the Basel Ganglia model, and then set certain meta parameters. Then in that meta parameter space, you can represent the patient. Because PD patients come in different subfamilies. There are patients in whom there is who are tremor dominant, patients who are rigidity dominant, patients in whom there is something called impulsivity control disorder or ICD. So the different sub flavors of PD. So we feel that if you extract these meta parameters and cluster them, these clusters will reveal the different subcategories of PD patients. And depending upon what cluster a given patient falls in, we should be able to develop a targeted therapy for that patient, right? And instead of using guesswork and some random thumb rules, we can let the model and the data prescribe what is the optimal treatment for that patient. So another line of work in the lab, which has been quite exciting, been happening for 5-6 years now, is modeling hippocampal spatial cells. So hippocampus is again a deep brain structure located inside the temporal lobe. It's been a field of very exciting research for a long time, for more than half a century. And two studies, particularly two studies on hippocampus, the one way back from 1970s. So this one was by John O'Keefe and his colleagues. These guys have found that so a lot of the experiments are done in rats and mice. because these animals are very good at spatial skills or spatial knowledge. So if you leave them in a maze, they can quickly learn the maze and find escape routes. So these people have found that. So they were recording from hippocampus from CA1 feed of hippocampus. You have seen that in a couple of classes ago. So when they're recording from CA1, they found that these neurons fire only when the animal is in one particular part of the maze, as though these cells encode that particular place in the maze. So therefore, they are called place cells. Then much later, almost 35 years later, in the Norwegian couple, Maybrit Moser and Edward Moser, they recorded from cells from entorhinal cortex, which is like the gateway to hippocampus. And there they found another kind of cells which fire not at just one location, but a whole bunch of locations. And what is interesting is all these locations form a nice spatial periodic pattern. And therefore, they named them the grid cells, which was very intriguing because brain is a very messy and disordered, uh, you know, uh, disorderly organized network of cells. How do you get? Because it's not like crystal structure. Crystals are very orderly, but neural arrangement in the brain are not. It's not very orderly. So how do you get this kind of orderly firing patterns in the hippocampus? So that was very intriguing. And now we have lots of theories and all, but when they found it first, it was very intriguing. So these two discoveries were jointly awarded Nobel Prize in 2014. So there's a lot of that created a lot of buzz and a lot of attention directed towards hippocampus since then. So this video shows what a place cell is. So here you see the wires stuck into the animal's brain. It's recording from a CA1 cell, and the experimenter is throwing some food pellets. Animal is moving around just to induce it to move around. Otherwise, it will just sit in a corner. Right, as it is going around, you see that the red dots indicate the firing events of that particular neuron from which they are recording. So whenever it goes to that part of the box, that neuron is firing. So it means that that neuron represents that location, that place. So we have taken up this study to understand how these grid cells and place cells form. So we made a simple hypothesis at that time. It's more like a good guess, but later on we found that there's external evidence. It's already in literature, but it's just that we didn't know at that time. So, for example, there are these oscillations called theta oscillations in hippocampus. The theta is in the range of 6 to 4 to 8 hertz. So these oscillations are influenced by the animal's motion. The theta oscillation frequency is influenced by animal's motion. Right, as motion increases, the theta frequency shifts slightly towards higher values, and not only that, the harmonics also pick up, right, as the velocity increases. So basically, the frequency is controlled by velocity. So we have taken up that point and proposed that at the input motion is represented by certain oscillations given by sin omega t plus some factor beta times integral of u dot v dt. Okay, so u is this. There's this cells called head direction cells, right? And which fire whenever the animal is going in certain direction, when it is going in some direction, is head direction. So we assume that the head direction cell output becomes the input to these oscillations. That's just a reasonable guess. And after that, we did some standard neural network theory, doing principal component analysis and things like that. And we found that that little trick gives you grid cells. So we got a lot of results, which published and described it in a couple of papers. Then we used the same model and applied it to 3D navigation, because this first model was for 2D navigation. A lot of experiments are done done in the case of rats and mice, navigating in the 2D plane. There are a small number of labs working on 3D navigation. One of these labs is headed by Michael Yartsev at UC Berkeley. So this particular picture is from our simulation, but Yartsev and Ulnowski, who was in Israel, right? These guys have done a lot of work on 3D navigation. So this is done in bats, because bats can fly. They're mammals, but they can fly, and therefore they have. Since they're mammals, they also have hippocampus. So in a set of very slick experiments, they record from a flying animal, right, from the hippocampus of flying animal, and there is a transmitter in the brain which transmits signals by telemetry, and so information is recorded. So they found that bat's hippocampus also has place cells. So we expanded the model to the 3D navigation case and found that we get place cells here also, just like what they found in the experiment. But our theory has thrown up two other interesting kinds of cells. This work was done by Karthik for his PhD thesis. So, so there is another kind of cell which fires whenever the animal crosses a certain, or the simulated agent crosses a certain plane, right, embedded in the 3D space. So we call this the plane cell. And there's another kind of cell in the model which fires whenever the animal, the simulated agent crosses not just one plane but a whole bunch of planes. Okay, so we called it the stack cell. It's like a stack of planes. So these two, at the time of publication, these two other kinds of cells were not known. But Karthik, after his PhD, joined Michael Yartsev's lab in Berkeley, and he's dug up the old data, and we found that there are indeed cells which look like plane cells. Then people have found grid cells also in response to eye movements, because normally grid cells and place cells they're thought to correspond to your physical motion of the whole animal in the external physical space. But when you move your eyes, that's also a kind of motion. I mean, it's also kind of a spatial navigation. It's just that your eyes are moving in the angular space of theta and phi. So even for that, people have found that there are grid cell responses corresponding to these eye movements, moving on some image space. These cells are also found in internal cortex. So we have modeled that also and found similar grid cell responses. So now Azra is using deep networks to figure out how to combine this velocity information coming from the locomotion apparatus, like our legs and movement of legs, and how to combine that with sensory information like visual information. And she's trying to explain a whole bunch of experimental studies. Then again, this Harsha, Azra, and we are also getting inputs from Dr. Ayan Mukhopadhyay, who is a theoretical physicist in the physics department here. We are developing a theory of 3D navigation. So what is interesting is it seems to be linked to a certain interesting body of mathematics called spherical harmonics. These functions arise in solution of Laplace equation del square equals 0. Right, Laplace equation solved in spherical coordinates. So if you remove the r part of the solution, the theta and phi parts of the solution is nothing but the spherical harmonics. These functions arise in the modeling of 3D navigation. So we're still working on this. We still haven't published this. So next thing that we are doing is modeling stroke rehabilitation. This is done as a project funded partly by government and partly by TCS. So stroke is a major killer, major cause of death after coronary artery disease. And so we're trying to combine computational modeling, neuroimaging, and virtual reality-based gaming. Our first model consists of bimanual model. So where we model the cortex at basal ganglia loop. Right, I talked about this part of it in my other basal ganglia talk. So we have made two copies of it to represent the two hemispheres, and we couple them. Because hemispherical interaction is very important. To understand it is very important. To understand stroke recovery, right? Because the two hemispheres seem to interact sometimes positively, sometimes negatively in in causing the stroke recovery. So we need to model like two hemispheres. So the model, one interesting question we asked is, in some literature, experimental literatures, they argue that, or they demonstrate that it is important to use both hands in your rehabilitation exercises. Whereas in another approach to stroke recovery, they tie off the normal hand so as to force the affected hand to move more, because only when you move the hand a lot, the hand will recover, the hand function will recover. But then the two approaches seem to contradict each other. So which is right? Who is right? Or rather, which approach to use in what condition, in what context? So that was a problem. So we asked the model of this question, and asked this question of the model, and so what we found is some kind of reconciliation between the two approaches. The model suggested that when the lesion size in stroke, because the stroke lesion occurs because of loss of blood supply to a given part of the brain. So when the lesion size is small, bimanual reaching same based rehabilitation is better than this constant induced movement therapy. So where you tie off the normal hand and force only the affected hand to move. So bimanual is better than CIMT in case of smaller lesion. In case of larger lesion, it's the other way around. This is what we kind of conclude from the model. This was published in scientific reports a couple of years ago. And now Sundari, my other PhD student, is working on use of simple deep networks to model visuomotor function. That is, input to it is visual input, stereoscopic input coming from two retinae, and output is a control of both arms. So now in this, you damage the layers close to the output are like motor cortex. Layers closer to the input are like visual cortex. So damage these layers close to the output, inducing some kind of a motor lesion, motor cortical lesion, and see, and then you try to retrain the network, which is like rehab, to see how to best gain the functions. Uh, so one of the things we found is that training the network on exploratory movements is better than stereoscopic movements. It is in traditional stroke rehabilitation, the patients are made to make very repetitive movements, which can be quite boring. So whereas there is people like John Krakauer in Johns Hopkins, he proposed that you need to make more extensive exploratory movements, right, and that will induce better rehabilitation. So that was given as a proposal or a hypothesis, and the network seems to be a recovery pattern of the network seems to be consistent with that proposal. We have a few other results. This was submitted, it is under review. So we also developed a comprehensive gaming system, right, which is now taken for clinical trials by Sri Chitra and Nimhans in Bangalore. So then oscillations, I've already given a, I think, long talk in this course on oscillations. I'll skip all this stuff. Then deep vision, some of the earlier work by Ryan Phillips for his PhD is developed a model of a retinotopic map. That is, how is the external space, right, mapped onto the primary visual cortex, the V1 space. So if you external space, if you model on a 2D plane, right, in polar coordinates, r and theta, right, which is for elevation and azimuth. So, so eccentricity and azimuth, and these two variables or parameters are mapped onto cortical space in a very strange fashion. So people have found empirically that this mapping can be modeled very well by a complex log function, the complex function, logarithmic function, but in the complex domain, which is very weird. Why will brain use complex numbers to come up with such a basic representation? So empirically that fit was known, but there is no theory to explain why it happens. So Ryan developed a simple learning algorithm which shows why or how that kind of a mapping develops. Then Anila used a network of 2D layers of neurons which have lateral connections. These are called lissomes, which are proposed by a group in UT Austin. So she has shown that these lissomes can learn direction sensitivity. Only thing is, if you have to train them using what is called asymmetric Hebbian learning. Right, in the Hebbian learning that we have seen in case of Hopfield network, it is symmetric because the weight update simply depends on the correlation between the presynaptic activity and the postsynaptic activity. But you take the activities at the same time instant, because right in the Hebb's rule that we have used, there is no time. You just take the pattern and calculate SI, and set the weights. But whereas in asymmetric Hebbian learning, you take the activity of the presynaptic neuron a little before the activity of the postsynaptic neuron. Okay, so there's a temporal mismatch. The idea is that it's a presynaptic activity that causes the postsynaptic activity after a little time. There's a time delay. So we have to take the two activities with the time delay. So when we do that, the network is able to learn what is called direction sensitivity. It will respond to moving stimuli. Like for example, if you have a bunch of moving bars, right? If the same bars with same orientation, but moving in opposite direction, will induce activities in different neurons. So then we also use such models to extract objects in so videos and images. So normally road signs from traffic videos are extracted from road images, but we thought that if you use a video instead of an image, you might get better accuracy, and we used a more temporal model to extract road signs. Okay, so this was done by Shweta and Neeraj. Then finally, we have been doing a lot of work on modeling the neurovascular coupling. This has been going on since about 2010. So like any other cell, neurons also receive energy from the blood supply, right? And so the blood vessels carry glucose and oxygen. So glucose is picked up by this intermediate cell called astrocyte, which is then released to the neurons. So we said that we asked this basic question long ago: why do you need these intermediate cells called glial cells or astrocytes? So we said that when the neuron receives input from some other neurons, the energy which is delivered by glial cells to the neurons, right? So we said that these two patterns must match. That is, neurons which are working very hard should receive more energy, and neurons which are not working hard should receive less energy. So the pattern of stimulation of neurons and pattern of energy delivery to the neurons must both match, because that's just engineering common sense. Otherwise, it's inefficient delivery of energy. So what happens if you assume that there's a lot of interesting biological consequences? You can develop learning algorithms for training the vessels also and the astrocytes, right? So you put all three cellular systems on a learning framework, which was very very radical at that time when it was first published. So then we developed a more detailed model of the neurons, astrocytes, and blood vessels. This was by Bankim for his MS thesis. Then subsequently, Karishma has developed a series of models simplifying them to show how you get a wide variety of dynamical regimes in this model as you vary your energy delivery. Then more recently, Bhadra and Nagavarshini and Soumya they proposed a model we called artificial neurovascular network. That is, in this network there is a deep network, right, and also a vascular network. So the neurons fire only when they receive energy from the vascular network. Vascular network forms a tree structure. So you need to decide which neuron gets how much energy, because all neurons will not be getting same energy. So how do you decide which neuron will get more, which neuron will get less? So just as we decide which neuron will be active, which is not, by training, by backpropagating error, we also decide how much energy is delivered to which neuron by backpropagating the error. So that is error backpropagated to the hidden neurons of the network. Is also propagated upwards over the vascular network, and you train the connection strengths within the vascular network, which is very interesting. So by this modeling framework, we have to view the vascular network also as a training system, as a trainable system, right? So you can think of information representation in the vascular network and things like that. So this was published in scientific reports just a few months ago. So we also proposed something called a flip-flop neuron some years ago in 2016. Then Vigneshwaran and Sujit have taken this and done a lot of runs. Even Shweta is involved, and have shown that this is better than LSTM on a lot of data sets. So long-term goal is to develop an oscillator brain theory or oscillations-based brain theory, and explain a lot of phenomena. This is a wonderful book by Georgi Bazaki called Rhythms of the Brain. Right? Basically, we want to come up with a computation model of the phenomena that he describes in this book. And we also want to implement this in a kind of an embodiment, like a robotic embodiment, because that will be a real proof of, like a demonstrator that all this theory is not just theory, but it works in a real case. So, like just to repeat, the long-term goal is to develop this kind of large-scale brain models and develop applications both in clinical neuroscience and in engineering domains. So, my dream is to create research centers. Ideally, this is how I think neuroscience research should work. The computation modeling should be at the core, and the model should be so versatile, such that they'll be able to take data from many different sources of neuroscience methods, like behavioral imaging, electrophysiology, and so on, and then make models which are consistent with all these different data sources, and then develop disease models, develop startups based on that, right, and then develop clinical applications. So this is a kind of long-term vision. Yeah, that's all. Thank